Okay, so hi everyone. My name is Momo Arai. I'm a fourth year medical student, and today I'll be teaching you guys about introduction to protein synthesis. Um, so first, wait. So if you guys have any questions throughout the lecture, feel free to let me know in the chat, or feel free to unmute yourself. It's fine. And I hope that this really benefits you, inshallah. So okay, let's start. Wait, no. There was something that showed. Okay, let's start. So, so here are the objectives, and I did the same exact lecture last year as well for PAL, and I was asking everyone what do these two things have in common. And just to start off with a fun fact, both of these have proteins named after them. Okay, that's it. But I hope that like there were lectures before this that talked more about transcription and translation of proteins, but this one talks more about how proteins are really made, and basically it focuses more on translation more than the transcription part. But do you guys have any questions about transcription before I go into the lecture, or any questions in general? Okay, if no, I'll start the lecture. So basically, you guys all know the central dogma by now. We have DNA in our bodies, which has the codes or the genes that basically. Oh wait, someone has questions. Sorry, go ahead, because I want to make
Salamat.
sure. Yeah.
Assalamualaikum. I just had a question about the RNA editing, specifically the site-specific deamination. So the one that goes from, I believe, cytidine to uridine.
Oh
So
yeah,
I
yeah.
was just, yeah, so I was just checking the slides, and I had a question about that. 1 second. Let me
Okay.
pull up the
I'll.
exact.
Yeah, sure.
So, mm, sorry, it's just.
It's okay. It's okay. But basically, what he's asking about is, I believe like the bases, when they're deaminated, when one of them is deaminated, it changes to another base. So bases are like they make up a portion of the nucleotide, which is, for example, thymine or adenosine or cytosine. Yeah, go ahead.
So basically, on this is about RNA transcription too. So
Yeah,
basically, just for reference, on slide 23, it says the C to U substitution involves a cytidine deaminase that deaminates a cytidine into a uridine. So basically, it says that a C is becoming a U,
yeah,
essentially. So
yeah.
could you just like explain more about that in general, and then based on that, I might have a follow up. So.
Okay. So sure. So this is for like, in case it helps anyone, I'll explain it here. So basically, do you guys know the four bases that make up the DNA or the gene, right? So it's, for example, for DNA, yeah, A, C, G, and T. So we have, they all have their own structures. A and G, yeah, you guys are both right. So for example, A and G are called purines, and C and T are called pyrimidines. But basically, cytosine has, you're not supposed to know the structure at all or memorize it. But basically, let's say the structure of cytosine. So that's C. And deamination, so it has a lot of groups around it, which is not an exact one, but this is just an example. So it has a lot of like amino groups, hydroxyl groups around it. Deamination just means that so from the name deamination, so you're removing an amine group. So this group is removed by an enzyme, which is, as you said, uridine deaminase, if I'm not wrong, or was it cyto? Yes, but it was like deaminase enzyme, right? So it basically removes the amino, like, yeah, NH2 group, and that's the structure that forms after that is the same structure as like uracil. So uracil is another it's another base that's only found in the mRNA or RNA in general. So it's not in the DNA, but it's in RNA. And just the removal of this group changes the structure. That's all it means. Is that clear for you and everyone?
Yes, I just have then a follow-up question about that. So basically it becomes deaminated. So basically the NH2, let's say, gets removed and or
Yeah.
whatever, something happens and then it becomes a uracil.
Mm-hmm.
But then I was just looking like at the line right above that in our notes. It says there are two types of RNA editing reactions that occur in the mammalian cells. One of them is the inosine for the adenosine, and then
Mm-hmm.
the other one is the substitution of a uridine for a cytidine. So I'm just confused,
Okay.
like what? So is it like the C is being substituted for the U, or like how's that
I
work?
yeah, I didn't exactly like go through the other lectures that you guys had, but what did it say exactly again?
So it says exactly there are two types of RNA editing reactions that occur in
Yeah.
mammalian cells. Number one, substitution of a uridine U for a cytidine C, and then two, substitution of an inosine I for an adenosine A. And then right below that,
Oh,
it says, yeah, right below that, it says the C to U substitutions involve a cytidine deaminase that deaminates a cytidine base into a uridine base. So I'm just
okay.
confused about like what
The,
which one is happening because they're contradicting.
okay,
Sorry
yeah.
for wasting your time if it's like
No, it's okay, it's okay. I'll try to finish the lecture. But I hope this point stays clear for everyone. But basically, yeah, the second point is this the point I mentioned about cytosine being deaminated to uracil or the uridine base. Inosine is I don't think you guys took it yet, but it's another like base that's not, that's not the typical kind of base here, but it can be changed adenosine. Now, you'll see inosine in the future lectures. I believe it was like, it's not here, it's not here. There's like a lot more. There's like xanthine, there's hypoxanthine. A lot of these nucleotides are made with this base. So you guys know how, for example, in DNA, so I'm talking about DNA. DNA has like a it's deoxyribonucleic acid. So it has deoxyribose, and then it has a phosphate attached to it, and it has a base. It has a base here. Now this base can be ACGT. There's another nucleotide that can form in later reactions, not here, not in DNA, but they're basically called, it can be called hypoxanthine or xanthine. And these bases, the base for that is inosine. So I hope that's clear, but I know it can be confusing because the doctor mentioned it in this lecture, but he just gave an example. So inosine can also be changed adding a reaction of its own. So that's the main idea. You don't need to like know the exact details of it because they will come later on in the lecture. Is that fine? Okay. I hope that's clear, but yeah, basically, these two examples are bases that can be changed into other bases by RNA editing. So they can change into like from one base to another based on the structure. Okay. Okay. So if you guys don't have any more questions, yeah, like I'll start with this lecture. Again, the central dogma is you go from the DNA, which is the code in our cells, and then you make mRNA, which is a copy of the DNA by transcription. And from the mRNA, you go to protein by translation. Now, transcription happens in the nucleus, then the mRNA goes out of the nucleus, and then it gets translated on ribosomes, which are the organelles in our bodies or in our cells where protein synthesis takes place. Now the proteins, so the proteins are made up of small subunits called amino acids. And basically the genetic code in this slide that he wants you to know is that three of these bases, okay, remember three as the magic number. So three bases here, or three nucleotides in like all of it together make one codon. And one codon, like one code, codes for one amino acid, okay? I hope that's clear for everyone, but this is the main idea of this slide. Okay. So I'll move on if there are no more questions, inshallah. Okay, so now there are a lot of, like I said, each 3 makes 1 codon, and 1 codon makes 1 amino acid. Each 3 here, you're not expected to memorize the whole, again, you're not expected to like memorize the codes for each amino acid, but basically these are mRNA. So mRNA, the only difference it has, one of the differences it has from a DNA is that it has uracil instead of thymine. So it has a U as one of the bases. Now, okay, so the main thing here, they have, so like the codes, the genetic code itself has a lot of properties, and the only thing that you're supposed to memorize or supposed to recognize in the exam are the ones that are highlighted. So AUG is always the start codon. Whenever, like all proteins that are being made start with AUG or methionine. But just if you see AUG, just know that it's the start codon. For stop codons, there's 3, and they're easy to memorize because they're kind of like they look similar to each other. So UAG, UGA, and UAA. So all of them have A, and then G and A just get like rearranged. So these are the stop codons, and this is the start codon. Now, you don't need to memorize that, but methionine and methionine and this is like they're the only ones which have 1 code, because as you see here, like for methionine is here, right? It has only AUG and it's the start codon always. But for example, let's see histidine. It has CAU and CAC. Now the third base can be different, and it's fine because it still codes for the same amino acid, which is histidine. And this property is called something called degenerate. So the genetic code, when the doctor says the genetic code is degenerate, that means that sometimes, even though the third base is different, it still codes for the same protein or amino acid, and it's fine. It's not a problem or anything. It's like normal to have that. The other point here is that the code is specific. So you see like each of these are specific to 1 amino acid and universal. So it's all over our cells. It's everywhere. The same CCU will always code for proline, for example. And yeah, so these are there are 64 different combinations you can have in total. Is everything clear from this slide? But the things I mentioned are the most important parts. Is it okay if I move on? Okay. Okay. So now we're moving on to more of like the problems that can happen. Do stop codons code for amino acids? Okay, yeah, so I'm coming to that point later, but basically you see here AUG is the start codon and it codes for methionine. So start codons will code for amino acid, but stop codons don't code for any amino acid. So if, for example, if ribosomes they recognize or they see stop codons, they won't have any amino acids attached to them, which I'll come to later. But I hope that's clear for now. So they don't code for amino acids, no. Okay. You're welcome. So now we're moving on to problems that can happen within the genetic code. So let's take serine as an example. It's UCA, right? And again, like I said, the third base can change, and that's fine. So it can sometimes be UCU because the transcription is not always it doesn't happen perfectly all the time. There can be mistakes done in our body. So if it's UCU, if the third base is the only one that's changed and it still codes for serine, then that's fine because it won't cause any issues, right? It's the same amino acid. So this mutation or this change is called silent mutation because it doesn't do anything. It's fine. Now there's two mutations that can cause a problem in our bodies. That's missense and nonsense. So if the first, let's say by mistake, it coded for CCA. Now the amino acid that's for CCA, like I said in the last slide, is now proline. So since the amino acid changed, it can cause a change in the sequence of how our amino acids are, and that's called missense. So because if the amino acid changes with the mutation, it's called missense because it doesn't, it's a mishap, like it codes for something else now. Let's say it becomes UAA. Now UAA again, it's one of the stop codons like we saw, and it does not code for any amino acids, but this mutation is called nonsense. So we used to memorize this by saying that by saying stop this nonsense. So if it's a stop codon, that's a nonsense mutation, and if it's the other one, which is change in amino acid, that's missense. Okay. I hope that's clear, but basically changing one nucleotide is called a point mutation because it's like only one point or one base that's changed. I hope this is clear for everyone. I'll give you more examples as I go. Okay. Okay. Now, other than changing the base, other than changing, let's say, a C to a U or C to any other base, you can add different bases or delete bases from the original sequence. So this here you see is the original genetic code. It codes for serine. Yeah. And so wait, let me answer, sorry. And then I'll move on. Nonsense are stop and missense are start. Yeah, it's okay. So nonsense are sorry. So if it's nonsense, it means it changes to a stop codon. Yeah, you're right. If it's missense, it's not a start codon, but it just changes the amino acid. Is that okay? Like it changes the amino acid to something else. It can be start, it can be in the middle of the sequence, but not stop. So if it's stop, it's the nonsense one. If it's any other amino acid, it's missense. Okay. Are the mutations dependent on the location? For example, okay. So if for example the error is on the third base, mostly it is silent mutation, and they will say in the again, you're not supposed to memorize this, right? But if you see here, even if you change this to a U, it still codes for the same amino acid. If it does, most of the time it's silent. But sometimes, for example, okay, so for example, this one, histidine, CAU, if this changes to C, it still codes for histidine, which is a silent mutation. But if it, for example, changes to A, it changes to another protein. So it's not dependent on the location. It can be missense, even if it's on the third. Okay. Okay. Now, yeah, missense, we're changing the whole amino acid to something else. We're changing histidine to this or this, anything, any of the amino acids. Is that clear for you? Okay. Okay, again, other than the missense, the nonsense, or the silent mutation, which are again all points because you only change one base, you can either add a base or delete a base. Now this is yeah, okay, it's fine. How are we supposed to know if it's missense or silent? Oh yeah, don't worry about that because usually they will give you, like they will, for example, I have a question coming up, this one, which we'll answer together, but they will have the code and they will have this, like this, they'll have the amino acid, and then the next code with the mutation, they'll have which amino acid. Don't look at the question yet. But yeah, is that clear? Like, you're not supposed to memorize, but they will tell you if it changed or if it didn't change. If it didn't change, it's silent. If it changed, it makes sense. Okay. Now, yeah. Again, okay, so this is the sorry, so this is the original sequence again, and then if you want to add bases to it, if you add, like if it by mistake adds a base to it, for example U, and it comes in this position here, and it inserts itself, you see that the way it's read now, since it's 3 by 3, now it's read as UCC instead of CCU because you added U here, so it reads it like UCC and then UAU like there. So it changes the whole sequence of the gene. And then even if you delete one, even if you remove this, now like if this is not here anymore, you would read it as CUA here, like here, CUAUGG. So the whole sequence changes with addition or deletion of 1 or 2 bases because it's read by 3, right? Now, if the whole sequence changes, it completely changes the protein. Like the protein structure is like abnormal now. Like it says here, 1 or 2 nucleotides added or deleted, it can cause frame shift mutation. So it shifts the whole frame. That's where the name comes from. But I hope that's clear. Is AUG only present at the start? Yeah, it's basically the start, like the start. Oh, no, sorry. So it can happen anywhere in the middle, but it has to be at the start. Because methionine is an amino acid, like normally alone. Okay. And yeah, so frameshift mutation, mutation is one of the most like dangerous mutations that can happen because you're basically changing the whole sequence, right? So it's not the same, not the same structure at all now. Do you guys have any questions from this? Because the question is, I have a question for you guys in the next slide. Okay. Okay, I'll move on. So so you have this sequence, and the one highlighted is the one changed down in this sequence. So what kind of mutation do you see here? I'll wait for more, but everyone so far is saying E deletion. Okay. Yeah, you guys are right. Anyway, good job, everyone. But basically, what I meant from this question, or the point that I wanted to get off, is that if there's 3 deletion or addition, you're taking the whole amino acid out of the whole sequence. So now there's no phenylalanine anymore. Instead, it just skips this and it goes to glycine, valine, serine. Now this does not cause frame shift, which is option A. It does not cause frame shift because it didn't shift the whole frame. Like all of these after the amino acid stays the same. It's not point because it's not one base. It's not nonsense because it didn't change the amino acid to a stop codon. It's not silent because it's just like it's not point mutation anyway. But then you're just basically deleting this whole amino acid. So that's an example of deletion of 3 bases. Okay, I hope this is clear for everyone. And I'll move on if it is. Okay. But yeah, that's good if you guys understand that. Now, this is an example of a missense mutation. Oh, sorry, someone was asking. Can there be addition of 3 base pair too? Yeah, there can be addition of 3 base pair. Just how you can have deletion, you can add 3 bases. But again, this won't cause frame shift. You're just adding like one whole amino acid to the sequence. Okay, I hope that's clear too, Chan. Now this is an example of a disease. Wait, sorry. Frame shift only occurs when half bases, yeah, 1 or 2, sorry, 1 or 2 bases are added. Yeah, exactly. So it's also mentioned in your slide by the doctor as well. So if it's 1 or 2 only, that's when frame shift happens. And you can see it by yourself, like when it's coded, when you see the amino acids, all of the sequence changes after the mutation. So that's why. Okay, now we'll move on, if that's clear, to this example, which is the sickle cell anemia. Now, this is an example of a missense, and it's important you guys know the exact mutation for the exam because they can ask you that. But basically, sickle cell anemia is a problem with the red blood cells when like there's a change in one of the bases, since it's point mutation, and it changes the whole shape of the RBC. It changes into like a sickle shape, which is like this, it's curved, right? So if it's curved, when it passes through blood vessels, it gets stuck in the small capillaries and the blood vessels, and that's why it causes all these symptoms. So this is what what the slide is, but basically you're not supposed to memorize all of these symptoms. There's a summary like here. If you see any combination of these symptoms like anemia, there's recurrent like infections, there's vision problems and pain, like bone pain, and they're feeling weak, that's when you know it's sickle cell anemia. Now, the importance of this, like knowing what the symptoms are or knowing the combination of symptoms, is that when they give you a case of sickle cell anemia and they say like, oh, he presented with this, and anemia, and what's the mutation causing the gene? So you guys know that this is sickle cell anemia that you're talking about. And the mutation is this. Basically, when we have the sequence, it's supposed to be this sequence with glutamic acid. Yeah, so don't get confused, because GLU is the glutamic acid, and the other one, glutamine, is something else, GLN, if I'm not wrong. So sickle cell anemia is when the glutamic acid gets changed into valine. Why? Because of point mutation. Again, one of the bases get changed by mistake, and it changes the whole amino acid. So when the amino acid is changed, only one, that's a point missense mutation. Now, this missense mutation makes a huge difference because glutamic acid is acidic. It has an acidic side chain. So if you see, it's like charged, right? Its structure has this. But valine is non-polar, which means it's like it's not charged, and it's like it stays away from water. So it like tries, when it's inserted into this like chain, it tries to hide away. Yeah, exactly, hydrophobic. So it tries to hide away from water, and like it folds on itself and so on. And that's why it causes not the disc shape that we normally have of the red blood cells, but we have like curved RBCs. Now again, make sure you guys know it's a point mutation in like beta hemoglobin chain only, not alpha. And then the beta chains are the ones that go like fold inside on itself. And yeah, if you want to know the position, it's position number 6, just in case. Again, this is what causes it to clump up in all the cells. Is that clear for everyone? Sickle cell anemia. The charged form of glutamic acid is glutamine, right? Yeah. Yeah. So sorry. So this one that's shown here is in the charged form, so you guys can understand that like what makes it different from the other one. So it's the same thing as writing COOH. So if it's COOH, it's glutamate, and then if it's in the ionic form, it's glutamate, sorry. So if it's COOH, it's wait, sorry. The charged form, yeah, the charged form is glutamate. Oh, I don't know why they switched up here. That's why I was getting confused too. But is that clear for everyone? So this basically is called glutamate. Yeah, you're right. Glutamate, but if it's the original form is COOH, is glutamic acid. But that's chemistry, so it's fine if just know that. Okay. Again, it's basically to do with the structure. So usually COOH is so you know how acids have like the full structure COOH, and then when it's dissolved, it becomes ions. So you don't need to know this, by the way, I'm just saying this for extra. But if it's in the ionic form, it's just called glutamate. If it's not, it's called glutamic acid. That's it. The name is just like different. Is that clear for you, Lean? Because there's another question. Okay. So which one's in the 6th position in the normal beta globin chain, glutamate or glutamic acid? Okay, so this is glutamic acid. And I don't know why. Okay, but basically glutamic acid and glutamate are the same thing. But if it's like dissolved, it would be in this state. If it's not, it's the other state. Is that clear? Like it doesn't matter if you say glutamate or glutamic acid. Usually we say glutamate, but the question won't have both of them, like for sure, because they're the same thing. Is that clear for you? Because someone asked me. Okay. Okay. So is sickle cell anemia clear for all of you? So the main important thing is point mutation, right? Of glutamic acid or glutamate into valine. That's the main thing. Okay. Now another mutation that can happen. So remember how I said how someone asked me about you can have addition of 3 genes. This is sort of like that, but basically you have a protein called Huntington, and it's in our like neurons, in our cells, like in the neurons, and the sequence is CAG, CAG, CAG. Uh, but sometimes by mistake there can be an addition of a lot of CAGs, so there can be an expansion of CAG a lot of the times, it's like an abnormal amount of times. Now this makes the chain much longer, and because this is the Huntington protein that's abnormal, the disease is called Huntington. And because it has extra copies of the same sequence, it doesn't cause any like frame shift or anything, but it's just an expansion of something that's already there, but in a large amount. Is this clear for all of you? I just added this, by the way. Oh yeah, like the picture, so you guys understand what I mean. So can we say it's addition? Yeah. But usually the term we call for this is trinucleotide repeat expansion, because you're adding it too many times, so it like expands a lot. That's why the name is like that. Okay. Now Huntington disease is a disease of like the symptoms will be things like they lose cognitive ability. They start to have like tremors in their hand because their neurons are like dying because of all these proteins. So once you have a long protein, it kind of again misfolds on itself a lot because the structure is abnormal. And then once it does, it aggregates into like these large clumps, and then it causes death of the neurons. Okay, that's basically it from this slide. Again, polyglutamine repeat, because so this sequence here, CAG codes for glutamine. So here, actually, it says glutamine, that's the amino acid, and glutamic acid is when it's like dissolved. But again, it's the same thing. If you want to be safe, just use glutamine. Oh no, wait, glutamate. Sorry, I'm so sorry. So glutamate, right? Yeah. This one is glutamine, and it's the other one, GLN. Is that clear for everyone? I'm sorry for confusing anyone, because I can explain again. Or we can write GLU. Yeah, you can. But usually, like, they won't ask you in the question, like, to write. So it's fine. Don't worry about this. So again, the part that because I was confused about this part. So glutamic acid is something else, and glutamate, because even before yeah. And then here for Huntington, it's glutamine, the other one, which is GL. It's not GLU anymore. It's the other amino acid. I'm sorry. So does mutations happen to all chromosomes here for Huntington? Or only brain ones? No, yeah, Huntington, like the Huntington protein is found in the brain. Like, it's, as far as I know, like it's found in the brain and the neurons. So that's why it affects these cells more than the others. Is that clear? Okay. Okay. Is anyone confused about what I said about glutamine and the glutamic acid? Because ask me now and I'll try to explain that again. Because I just got I just got what I said. Okay, if it's clear, I'll move on. And you can stop me anytime if it doesn't. So we talked about sickle cell anemia, which is the point mutation, and then missense. And then the second example we talked about was the Huntington disease, where you added a lot of trinucleotides. So it was an expansion, like repeat trinucleotide expansion. The third example that I want to talk about is CFTR protein, which is which is cystic fibrosis transmembrane conductance regulator. So I'll go over this point and I'll repeat because someone was asking me to. So CFTR is a protein that's found in our cell membranes. Usually it's a channel protein. So its normal job is to like pump out chloride ions so that water follows out, and then it's like kind of like it doesn't make the surface dry and mucus doesn't build up. But sometimes when there are mutations, so for cystic fibrosis, the mutation is deletion. So this is the question that I showed you before, but basically there's a base here, phenylalanine, at the 508th position, which gets deleted. So you guys answered all of this. This is deletion, and it gets removed from the whole sequence, and that's what causes this channel to stop working. So chloride ions don't go out anymore, and there's no water that follows it. So mucus builds up because now it's a dry surface, like there's no water. And then this mucus kind of like it blocks the airways, it blocks channels, and then it can cause like disorders of like the pancreas, intestine, and the lungs. Is this clear before I explain the other part, the glutamine? So this mutation is deletion. Okay. I hope this is clear. And okay, someone asked me to repeat this one. So for Huntington, just so you guys don't get confused in the exam, there's two different amino acids. There's glutamine with an N at the end, and there's glutamic acid. So glutamine, I memorized it because it had N. It was like when you write it in the short form, it's not GLU, it's the other one, GLN. But then glutamic acid or glutamate here, which is for sickle cell anemia, is GLU, like the code for it. But basically, these are two different amino acids. Is that clear? Yeah. So Sidra summarized it like perfectly. So glutamic acid or glutamate in beta globin for sickle cell anemia, which is point mutation, missense mutation. Glutamine is in Huntington protein. So sickle cell anemia as point mutation, but Huntington is addition. Yes, Huntington, instead of addition, they say more of trinucleotide repeat expansion just because you're adding a lot, but it's basically addition, like you're adding too much. Is that clear for everyone? Okay, so normally chloride exits the cell through CFT. Oh, okay, so this is about CFTR. You guys, okay, so just so, because I'm moving between the diseases, sickle cell anemia is again point mutation. Huntington is you add a lot, so it's a repeat expansion of glutamine. And then CFTR is deletion. So you're deleting a whole amino acid, which is phenylalanine, something completely different. So normally chloride exits the cell through this protein and increases osmolarity outside. So water follows by osmosis, making mucus less sticky. So it does not clog the airways. Yes, you're like absolutely correct. For the exam, is that clear for you while I read the next question? For the exam, do we need to know the exact protein or no, and like which codon it starts? Okay, you need to know the proteins that are abnormal in each of these diseases, but not, like only for the disease state, like not every protein. And codon which codon is start and which is a stop. Yes, you need to know that one only. So AUG is always start, and the other UAG, UGA, UAA are stop. Okay, so did everyone get CFTR, cystic fibrosis? The problem with cystic fibrosis? Okay, so I'll move on. Do you guys have any questions? Because I'll stop, any more questions, because I'll stop here for a bit If you guys don't have, I'll move on. Okay, that's good. Now, this is like the main bulk of the lecture, which is translation. So everything that I mentioned before was like diseases or mutations that can happen within the gene sequence. So for translation, you need mainly four things. So you would have you need the mRNA from the DNA. So mRNA is a copy of the DNA. It carries like the code from the nucleus. Again, it goes out to the cytoplasm. And then you need a ribosome. Again, like I said in the beginning, ribosomes are the place where proteins are made. And then the protein, like mRNA, comes to the ribosome, and then tRNA, which is another protein, joins them because it needs to bring an amino acid. Okay, and then the fourth thing is like you need enzymes for an energy for that to happen. So I'll go over that. CFTR is considered frameshift mutation. Okay, no, because you're removing 3 bases from the whole sequence. So it doesn't change the sequence of the other amino acids because it's read by 3. Yeah, so it's just deletion. Okay, so all amino acids. Okay, so this just it's a slide that just says about like you need all amino acids in your diet to be able to make all the proteins, because as we know, there's only 20 amino acids we have. So RNA. I'll go over the different types because like briefly, because I know you had a lecture on this before too. But basically, we have 3 different types of RNA that are important in translation. So we have messenger RNA, which is again the copy of DNA. Ribosomal RNA, from the name, it makes up the ribosome, which is where protein synthesis takes place. And then transfer RNA, it carries the amino acid. It carries amino acid from the cytoplasm to the ribosome, so that the amino acids can start binding together and make a huge protein or polypeptide. At which point does the mRNA bind to the ribosome? I'll explain that, but that's basically the first step. So mRNA has to bind to the ribosome for all of it to come together. And I'll explain it now. So this is how it should look like. And I honestly wanted to like draw out a slide, like draw it on a slide for you. So in case it makes it easier, but basically, sorry, okay. So basically you have the mRNA that's from the DNA during translation. Yeah. So we're not talking about translation because the mRNA is already transcribed the DNA from the nucleus. DNA. Now it's a copy and it moves out into the cytoplasm. And then we have the ribosome, we have a large subunit, we have a small subunit. As we saw in the picture before, it's just made up of 2 subunits together. And yeah, so this is the large one and this is the small one. The large one so this is a summary of all the slides that I'm going to talk about now. So the large one has 3 different sites, and I'll come to that in a minute, but it's like it's called A, P, and E, and it's like A spelled backwards like that. Yeah, but each of these have different like functions and they're important for the translation part. Doesn't the small one have those sites? Oh no, it's mainly the part of the large ribosomal unit, because the small ribosome is important for something else, which I'll say like right now. So you have the tRNA. The tRNA, I can't draw it, but tRNA looks like a hairpin loop. It's if you saw on the other slide, so mRNA is this genetic sequence. tRNA is something that looks like a loop because it carries amino acid on its end. So this is the amino acid. It could be the first amino acid is always methionine, and then it could be others like valine. All of it is specific. So each tRNA molecule is specific for its amino acid. So it carries a specific amino acid based on the structure. And then at the end of the tRNA, so one end has the amino acid and the other end has the code, like these are called anticodes. Okay? So the tRNA structure is made up of bases too. It's made up of bases just like mRNA, and it's at the end of it, it has 3 here that matches the one on the mRNA, which are called again we said they're called codons. So that's why the name is like anticodon and codon. 3' end is that, yeah, you're right. So 3' is here, where the amino acid is, and 5' is here. mRNA, from the transcription lecture, it's 5' here and 3'. So you can see that they're anti-parallel, right? Because 3 is here, basically, and then 5 is there. Then 5 is here, 3 is there. So this joins here. This is not, I'll make it more clear later on. But the anticodon basically sticks to the codons. Okay, now I just want to mention, so these are the things that we need for translation: mRNA, ribosome, and then tRNA. And then later on we'll have like energy and so on. But basically from mRNA, since you guys know the structure a bit, like you're familiar with it, you guys know that in eukaryotes and prokaryotes, they're the same, they're kind of the same, except that eukaryotes have a cap here, they have a cap here and a tail here. It's called poly-A tail because it has a lot of A. And the cap is guanosine cap. Basically, their function is to protect the mRNA as it goes from the nucleus to the ribosome. And the cap has another function, which is recognition. So who recognizes the cap of the mRNA? Because I said it before, it's the small ribosomal subunit. Again, now before the ribosomes are like stuck together like this, this is kind of separated away. So let's put this away for now. We have the small ribosomal unit here, right? And one part of the small ribosomal subunit recognizes the 5' end of the cap. So this comes here. Now we have the cap binding to this place. Now mRNA is here now. The small ribosome. Now since that's the first step, like I said before, recognition is the first step, and this is the initiation process. Once it binds, AUG would be here. AUG is always the start codon. So AUG would be there. And then this binding of mRNA to the ribosome is brings the large subunit. And it brings tRNA. Now, once all of these bind together, okay, it will look something like the picture I showed you in the last slide, but basically like that. So this is P. You have the small one here, and you have mRNA here, right? Now you have, I hope this is clear for everyone. If not, let me know, okay? So the tRNA, the first tRNA to enter the large subunit, so mRNA binds on the small subunit, ribosome subunit. tRNA comes and binds to one piece. Only the first tRNA for AUG, which is the start codon, comes to P. Okay, comes here. This is where the tRNA is. And like I said, this is the anticodon for AUG, and this is AUG. So they bind together. And remember how I said at the end of the tRNA, you have an amino acid. So it carries an amino acid with it here. It carries an amino acid. So you have one amino acid, and because the anticodon is bound to the codon now. Now the next tRNA, so the sequence continues here. It goes on. And then the next tRNA comes on the A side. So the first tRNA always for start codon goes to P, but every other tRNA that comes goes to A. So A is the first letter of the alphabet, so every other tRNA goes to A first. And then I'll explain from there, but this is how initiation and then elongation starts from here. Okay. I hope that was clear, because I just wanted to give an overall general idea. So the small ribosomal subunit binds to the cap or AUG. Oh, okay. Again, I forgot to mention that part because I drew it. I drew eukaryotes only. So eukaryote has cap and the tail. So small ribosome binds to the cap. It recognizes the cap, but the AUG is here. Since you guys know the structure is like that, right? So it generally like binds to the whole like ribosome, but it recognizes this first so that the whole mRNA can go on top. Can you recap again? Okay, I will throughout the following slides because this is just me like drawing it out. So slides might make it clear. So prokaryotes have a different sequence, right? Does anyone know what, there's no cap, but there's something else for prokaryotes. Does anyone know what it is called? What it is called? Or from your other lectures? Because there's no, yeah, you're right. So this is called Shine-Dalgarno sequence. Instead of the cap for prokaryote. Yeah, yeah, it's a fancy name. And then the tail, there's no tail here, but it has UUU instead of AAA. It kind of looks like a loop again. Okay, so that's the difference between eukaryotes and prokaryotes. Now I'll move on to the main part. So this was like the initiation part because I know it can be hard to understand that at first. But tRNA, you guys know now, it has a hairpin loop. It carries amino acid. Each amino acid is specific to it. And you don't need to know that, but there are a lot more tRNA than we have amino acids. So even if the last base of this is different, it can still carry the same amino acids, same as the mRNA that we mentioned before, the degenerate code. Again, this is how the tRNA looks like. This is a much better like picture. So someone said that the 3' end is where the amino acid is bound. This is the first one, methionine, and this is where the first one is here. So anticodon is this structure that binds to the first codon AUG of mRNA, and the amino acid is stuck there. It's just there. So that when you translate it, the amino acids start sticking together to make proteins. Is that clear for everyone? This is just the tRNA structure. It carries two things, anticodon to bind to codon, and then amino acid, which is coming later. Okay. Is that clear for everyone? Yes. The first tRNA is on the P side. So this will make it more clear. This picture, I have a picture coming up, so that will make it more clear. Okay. So tRNA again binds to the mRNA by anticodon codon. They're complementary, and the mRNA is 5' to 3'. So it's read by that, and anticodon is the opposite. It's flipped, so 3' to 5'. Yeah, here. So mRNA is 5' always to 3'. tRNA is 3' to 5'. Now, like I said, even if the last base changes, it's fine. It can still bind to the same codon, and it can still carry the same amino acid. So this is called wobble hypothesis. It's just the same name, like it's just the same thing, but the name is like given differently. Is that okay? So the small ribosomal subunit binds to the 5' methylated guanosine cap located upstream and reads. Yeah, you're right. So the cap is where recognition happens, and then it reads along the whole mRNA. Silent mutation? Sorry, wait. Oh, you mean silent? Yeah, for this one. Yeah, it is kind of like the silent mutation, but wobble here, it just means like the tRNA's anticodon. So the name given is just wobble hypothesis, but you're right, it's the same thing. Wobble hypothesis only during translation, and silent mutation is before. Yes. So silent mutation is in the code itself, like in the DNA code, for example. If it changes, it's silent mutation there. Okay. Now, again, this is the same thing that I tried to draw out for you. So this is AUG, start codon of mRNA, and you see it here, right? This is the ribosome. See how they're separate before it joins? But once it joins to the small one, the large one comes and joins them. So the large one comes here, and then you have tRNA, and slowly protein, like amino acid, again, join together to make a polypeptide, which later joins together other polypeptides to make a huge protein. And UGA is the stop codon where everything just like falls apart. It stops coding for anything, stops the synthesis there. Why are these circles? This is just the shape of, he meant like this is a protein that's like folded, and it's the same thing. It's just a protein. It's okay. And then, so ribosomes, something I didn't mention in the previous slides is the ribosome that's different from prokaryote and eukaryote. So prokaryotes and eukaryotes have different subunits. So total the S just stands for like the size of the ribosome. Both of them have large, both of them have one small and one large. But the number or the size of each is different. So prokaryotes have 70S, eukaryotes have 80S altogether, like the large and the small. Now, you can know that prokaryotes are like bacteria, so they're smaller, so it's 70 instead of 80. 80 is bigger, it's a bigger number for eukaryotes. For eukaryotes, if you separate this large subunit from the small, the large subunit it's a size, or like it weighs around, it's like given the name 60S according to its weight. The small one is 40S. Prokaryotes in the bacteria, the smaller one for 70S, when you divide it, it's 50 and 30. Should we know all the subunits and the, yeah, I was going to say that just now, because he told us we should know each of these subunits that make up each of these RNA that makes up each subunit. Just because in case they ask you like, oh, this was bound to 23S RNA, so the question can ask something like that. Which part of the subunit is it? So it's, if I say 23S, it's part of the 50S subunit of prokaryotic ribosome. Did it actually come in the exam? In our exam, thankfully, no. But that's what he told us, and it's safe to know it because there's a point where I want you guys to know what 23S RNA is. You can only memorize the ones that are important. Like, again, I keep mentioning 23S because that's where the enzyme is later on. But yeah, generally, I didn't memorize it, but like I hope it doesn't come in the exam because there are like many different topics that are more important here, more important to focus on. As long as you know the general idea, you're good. Do we have to memorize the number of proteins? Oh no, these proteins don't need. We never got anything about them. 5S, yeah, 5S. Just know for 50S, it's made up of 5S RNA, 23S. For 30S, it's 16S. Like you don't need to focus that much on it, but let's say, yeah, 5S is in both. You're right, 5S is here and here. So basically don't memorize it. It's in both. So you don't need to like worry about that. But if you want to know the only difference between these two, it's the 23S for 50S and the 28S for 60S. Again, for eukaryote, each of the numbers are just bigger than these. So it's kind of okay, like 23S is greater than 23, 28 is bigger than 23, and for the small subunit, it's 18 instead of 16. That's it. That's all I wanted to know. The main important thing is that when you see 70S, just know it's prokaryote and it's divided into 50 and 30. For eukaryotes, it's 80 divided into 60 and 40. Okay, is this part clear? Okay, that's good. Now, again, this is the same exact thing that I was showing you earlier. Small ribosome subunit is where the mRNA attach, mRNA binding site. Large ribosomal subunit is where it has all these sites and it has the enzyme that basically helps in the peptide formation or protein formation. Okay, so it has, since it's large, it's where the enzyme is. That's just memorize it like that. And that's basically it. So initiation, like I said, it first recognizes the thing, like whatever sequence is at the beginning. So this example here that's shown is only for prokaryotes. So prokaryotes, as someone mentioned, it has Shine the Garno sequence, and the tail is, yeah, it's not written here, but basically the tail is there's no tail. It's a different sequence than the eukaryotes. So let's focus on prokaryotes, but it's the same process for both of them. Prokaryotes, the small ribosomal subunit recognizes this Shine-Dalgarno sequence. Which part of the small ribosome? The 16S rRNA. That's why I was saying it's good to know them. So the small ribosomal subunit of prokaryote has only 16S. So it's fine. You won't get confused with any other like RNA there. So 16, whenever they say 16S, that's the small subunit of prokaryotes. That's where it's complement. So that's where it recognizes the SD, Shine-Dalgarno, which is located, as someone mentioned, upstream of the actual start codon. Now, this recognition starts the whole process, and that's what it means by saying facilitating the positioning of the small subunit with the start codon and the large subunit joins in. The doctor said it looks like it, but that is not the case. Wait, looks like what? Sorry. Okay, you can ask me if you have questions, it's okay. So initiation codon, again, is AUG, don't forget that. And the tRNA comes, the first tRNA for AUG comes on the P side. Now, the next, so once it continues, the ribosomes will start reading the mRNA on this, from 5' to 3' in this direction. So it will continue reading the sequence just so it can make the proteins. Okay, it looks like the EPA are a part. Oh, okay, it looks like the EPA are part of the small ribosome. Yeah, it does look like it, but like the general structure, do you see it better in this picture here? They're more part of the large ribosome. Like it's kind of like the shape of the large ribosome. He said the A, P, and E are actually on the. Oh, he did. I don't know about that because I always know that they're on the large ribosome, but I'll check. I'll check for that and let you know. Okay. Someone said Shine-Dalgarno is transcribed but not translated. Yes, you're right. So Shine-Dalgarno, the cap, the 5' cap for eukaryotes, they're not translated, but they're transcripted from the DNA. Yeah. So I think that like what you said is right. So it does look like it's on the small one, but it's on the large one, if I'm not wrong. That's what I know. Again, here it shows 2 in the picture. So I hope that's clear. And then, yeah, so first tRNA comes to P. Every other tRNA comes to A site. And then you see this protein that's coming out of the P site. Whenever the tRNA brings a new amino acid, so you have the P and the A, the amino acids that are stuck to P, they move, if you see on the slide, they move, all of it shift to A. And then both of these shift together to the other side so that this tRNA is now on the E site, which is exit, and the one on A previously is now on P. So the one on P will always have the amino acid sequence attached to it. And then when another tRNA comes on the A site, it will shift, the amino acids chain will shift to the tRNA on A site, and then both of these will shift together just so it repeats the whole process again. Is this part clear for everyone? Because this is the main step for the elongation part of translation. Just tell me yes if it's clear. So just so I know. If it's not, ask me. It's okay. Okay, I'll repeat it. So okay. So remember how I said tRNA. So once mRNA binds here, tRNA comes here. Okay. tRNA comes on the P site because it starts. AUG is the start codon. It's on the P site. The next tRNA that recognizes the sequence on the mRNA comes on the A side because that's the only space left for entry, So it comes on the A side. The amino acid on the P tRNA Do you want me to draw it out for you? Like, is that better or than explaining on the picture? Where did it go? I hope so. I'll try to make it quick. I know you guys have another lesson after this at 7. So again, the only function of the E is an exit. So the one you need to understand is the P and the A. Can you please explain the Shine-Dalgarno? Okay, I will. Okay, just a second. So this is how it looks like now. Okay, mRNA here, we're talking about prokaryotes because that's what the example he gave. So mRNA comes here on the small ribosomal unit, and the AUG is now here. Start Shine-Dalgarno, for someone asking, is here. It's not being translated. There is no amino acid for it, but it just binds to the substrate just so it can move the whole mRNA along. Right? So this is 5. Now, tRNA, the first tRNA, again, I'll keep repeating this for AUG, comes on the P side. So this is where the anticodon is for AUG. Okay. This is the first amino acid, which is methionine. Okay, methionine. The second, and all of the tRNA that comes after it comes to A, because now A is like the first letter. So that's A is where everything else comes first now. So this is where the second tRNA comes for the second sequence on mRNA. So that this joins just like this one, just like the next, and it just goes on. So tRNA comes here, right? The second tRNA. The second tRNA has its own amino acid here, specific to this code. So now you have one tRNA here and one tRNA here, right? This amino acid attached to this tRNA moves here. So that, okay, so this amino acid moves, it breaks off here and moves to the amino acid on A. So now the amino acid looks like this chain. Okay, this is A, this is P. I'm sorry, it's like squished, but that's how it is now. So do you see like how the amino acid on the P moves to the A to be on top of the amino acid on A? This sequence, this is now making a sequence, right? So we have one amino acid, two amino acid, and this process keeps happening so that at the end you have a lot of amino acids together like that into a protein. Is that clear for everyone? This moving part, it just moves, the amino acid moves. Okay, thank you. I hope so. Now, when it moves here, you see that the tRNA that was here is now empty. It does not have the same amino acid, right? So now, let's see it again. So the one on P, okay, now moves, the one on P now moves here to E because it wants to exit. It doesn't have any job anymore. It doesn't have any amino acid attached to it. So it moves to E. So the one on A, like with it together, moves to P because now there is space. So both of them shift. So now the E is occupied and P is occupied. The A is empty for other tRNA to come. So E is the one without amino acid, and the one on P is the one that we said here, like this one, and this is in blue. Imagine this. Okay, so this is the same thing here. It just shifted to P. So this is E, and A. Both of these shifted to the next available space, and the one tRNA here just exits from E. So it just goes back into the side. The P stays here. It needs the same thing happens to these two. So whenever the third tRNA comes with its own amino acid, these two shift again to this one. So it cuts off here so that it's now read to like the amino acids. Is that more clear for everyone? I hope so. But if it's not, I can re-explain it. I don't mind. Wait, what about the rest? Because I hope like this made sense, honestly. Okay. Oops. Okay. I'll clear that now. But basically that's the whole thing the picture is talking about. We talked about this. Shine-Dalgarno is just a sequence it recognizes and it continues. So yeah, that's this picture. So these amino acids here gets cut off and it moves to this. And then both of them shift together to like the one without amino acid exits and the one with the chain stays on P for the next one to come in, and it continues. So at the end, we have a lot of chain of amino acids. Okay, I'll move on because there's but I hope it's clear for everyone else. Basically what I showed you just now it's called elongation. So initiation was recognizing the mRNA, right? So is the mRNA moving or the ribosome? I mean it's the same thing, but there are instances where the doctor says, for example, the ribosome reads the RNA. So it's kind of the same thing, like the mRNA moves through it or the ribosome reads through it. It doesn't make a difference, you know? Like, does that make sense? We say that in biochemistry, like they say that the ribosome reads mRNA. So I always imagined like the mRNA goes through the ribosome and the ribosome reads it. There's one more example I'll show you, which might make it more clear. But elongation is the same thing that I mentioned. So ribosome moves from, here it says ribosome moves on the RNA, like ribosome reads the RNA. The enzyme that I forgot to mention, so that you see the amino acids, I erased all my drawings, but the amino acid when it here, okay, when it cuts off and it binds to new amino acids on this side, the enzyme that does this is peptidyl transferase 23S rRNA. The enzyme name is important. Does anyone remember where 23S was on the ribosomal subunits? Just know this one if you want to know anything out of large, large 50S. And when you say 50S, that's prokaryote. So you're exactly right. Yeah, big one of prokaryote. Yeah, you guys got this point, so I'm really glad. 50S, yes. Yeah. So here you go. So 23S RNA in the large ribosomal subunit 50S of prokaryotes. This catalyzes the reaction. That's why it's called ribozyme, because it's made from like an rRNA. That's why the name is ribozyme. And then this is known as translocation. Elongation, you can call another name for it as translocation. Now termination. The reason why I'm considering prokaryotes only is because that's the example he gave you on the slide. But generally, it's the same process for all of it. So if it was eukaryote, the enzyme would still be peptidyl transferase, but on the subunit 28S, which is ATS, which is the eukaryote one. Okay. So peptidyl transferase. Yeah. Did I answer you just now? For eukaryotes, it's in the 28S of ATS. Wait, I know it's like a lot of names and everything, but here 60S, sorry. So 60S is the large ribosome, and here 28S is the enzyme for eukaryotes. So the one we were saying just now, the prokaryote one, is here 23S of 50S. But if you're talking about eukaryotes, it's the same enzyme, but for this. In the picture, the APE are on the large ribosome. Yeah, you're right. That's what I meant. So it's on the large, from what I drew, it's on the large one. Is that clear for you? The one who was asking about eukaryote, like the enzyme. Let me know if it's not. It's fine. Okay. The largest RNA. Yes. So remember how I said it's large, so it has the enzyme to like make this happen. That's just it. So the large one has the enzyme too, as ribosome. We'll move on to the last step if you guys don't have any more questions, which is termination. So we said initiation, recognition. Elongation is when it moves the whole shifting from A to P to E is elongation. And then once it reaches the stop codon here for you, either any of these three, we said that it doesn't code for any amino acid. What it does is the release. So what comes to it is not a tRNA, but it's a release factor. It's just a release factor to help release the whole thing. So we don't want to, we want coding now for a protein. This is the complete chain of amino acid proteins, right? So it's done. Now we don't want any more. So the release factor comes in the A site as usual, but now there's no shifting that happens. The release factor binds to like an epithelial and it just makes the whole thing like dissociate. So it just breaks the whole thing away. So that you have your protein here, you have the complete here, and that's it. That's what you needed from this whole unit. So now it's gone. It waits for another mRNA, and then the whole process starts again. So termination is easy to understand, right? Is it okay? Okay. So termination occurs when one of the three subunits come, and then it moves. The release factor moves onto the A site. That's the whole. That's the whole slide. What I just said. And then it helps to release the whole thing. Now polyribosomes, there are some, okay, there's no picture for it, but there are some mRNA that's a very long mRNA. So at the same time, you can have one ribosome subunit making protein out of it. You can have another one making once this finishes, it goes into another ribosome subunit to start making again. And then so do you get my point? Like going inside, RNA is going inside, then another ribosome attaches to it. Yeah, okay. That's called polyribosome. So at the same time, you can have many ribosomes attached, and you can have many proteins made at the same time. That's the main idea of polyribosome. That's why I said I don't know. It's the same thing, but basically ribosomes like read the mRNA, translating the same mRNA into polypeptide. Yeah. So you have the same protein at the end. Now the last part of this lecture is, I'm sorry, I'm taking too much time. The last part of this lecture is protein targeting. Okay, wait, I'll stop for like the azan because you can hear it. But I'll start in like 2 minutes once it finishes. So we'll start from here and we'll talk about this. Okay, I will explain it. Just give me 2 minutes until the prayer, like the azan. Okay, I'm sorry, yeah, I had to pause. But someone said to repeat the termination part. But basically, termination is when you have stop codon come here, and instead of a tRNA, like for the whole process again, you have a release factor. Release factor from the name, when it attaches to the A site, it just breaks the whole thing so that it releases the protein here. You see here? Is that clear for you, Rida? So you just want the protein now, because stop codon basically just stops the translation. Okay, but it is, but 60S isn't. Oh, yeah. I know. So he goes between this and that, but if it's prokaryote, it's 50S. Okay, that's what I meant. So I know. Yeah, 50S. You're right. This is, by the way, exactly from what he wrote in the slide. I just added this picture. No problem. Okay, so last part of this lecture, inshallah it won't take long, but this is basically protein. After you have the protein, it can go and have like modifications, or it can be targeted into other organelles. So for example, this is an example, but endoplasmic reticulum is where all these changes happen. You can add a sugar molecule, you can add hydroxyl and all that. So for it to go into the endoplasmic reticulum, because we said ribosome is in the cytoplasm, you see this protein that's coming out of the ribosome. It's being made right now, but there's something called the signal peptide at the beginning, where it's recognized by signal recognition particle from the name signal peptide and the signal recognition particle. It binds together. The signal recognition particle like pulls down the whole protein so that it goes down to this like this kind of channel or opening, and then you see this part here. It brings down the whole protein or the amino acid chain as it's being made into the endoplasmic reticulum. Is this clear? Is this slide clear? It just brings it down, okay, to the endoplasmic reticulum. Okay, I'll repeat. So proteins, they have to like they can't stay in the cytoplasm for a long time. They have to go somewhere, right? One of the places it has to go is endoplasmic reticulum, which is another organelle where proteins can be modified. Now, once you have there's something in the cytoplasm called signal recognition particle, which binds to a signal peptide on the protein so that it recognizes it and says that, oh, I need to take this protein to the endoplasmic reticulum because it's like it needs to be changed more, it needs to be folded more and like joined to other particles. So now this signal recognition particle binds the signal peptide, which is on the protein itself. It brings it down and it just allows it to go through a channel into the endoplasmic reticulum. That's it, like that's its job. It goes away again, you see, like it goes to take another protein. Is that clear for you? Okay. I hope that's clear for everyone else. If not, let me know, it's fine. But basically, these signal peptides, these signals you see here are the sequence. Okay, they're called, so it depends. So usually they're the N-terminal hydrophobic signal sequence. If they're going to the endoplasmic reticulum, rough endoplasmic reticulum. Now, just remember that name because I don't know, it's the only it's the only thing you need to know. The other one is easy to memorize. It's called nuclear localization signal. So from the name, where does it go? It goes to the nucleus. So the protein goes to the nucleus. Okay? Does that make sense for everyone? So if it's rough endoplasmic reticulum, the signal sequence is just called hydrophobic N-terminal signal sequence. Okay? If it's nucleus, it's nuclear localization signal. Is it okay if I move on from this slide? Okay. Now, these are the modifications that can happen. Sometimes proteins can fold on its own. It can change, do all these changes on its own. Sometimes it needs the help of other proteins that are called chaperone proteins. From the name, they kind of help the proteins become modified until it's like the full structure and function. It has the full function of that protein. So that's the main thing from this slide. It's just that some need help by chaperone proteins. Now, the changes that can happen to the proteins are four main things, and that's like I'll end the presentation with that, okay? But I hope everything else is clear before it. So this is after it's being translated. After you have the protein, you can have either phosphorylation, which is addition of phosphate group. You can have glycosylation, which is addition of carbohydrate groups, or you can have addition of hydroxyl groups, which are OH groups, called hydroxylation. And the fourth thing is trimming, which I'll come to. So these attachments, the main important thing you need to like memorize from this is the amino acids that undergo all these changes, because they're different for each. So for example, the hydroxyl groups of serine, threonine, and tyrosine, the two T's and the serine for S, these are for phosphorylation. Okay, so that means that phosphate groups can be attached to the proteins, amino acids. So this is serine, and this is tyrosine, and phosphate groups are attached to it. Now, the way a phosphate is attached is by protein kinase. So kinase, as you all know, kinase adds phosphate groups to things. But phosphatases, so you see from the name phosphatases, it breaks down or separates phosphate groups from the amino acid. So to add it, kinase, to remove it, yeah, exactly, dephosphorylation, thank you. So dephosphorylation is by protein phosphatases. The three amino acids again, serine, threonine, tyrosine for phosphorylation. That's the first one. Second, glycosylation. The amino acids that undergo glycosylation are serine, again, hydroxyl groups of serine, threonine, or hydroxylysine. Now, serine and threonine are repeated from this one, so you can memorize them together. So serine, threonine, the first two here is the same as the first two for glycosylation, serine, threonine. The addition, like the new thing here, is hydroxylysine and asparagine, which I'll come to. So serine, threonine, hydroxylysine they undergo something called O-linked glycosylation. So glycosylation have two types, so O-linked and N-linked. So you can see here, for example, serine has an O-linked, and it's linked to a carbohydrate chain. So O-linked glycosylation. And this substance or carbohydrate is called N-acetyl galactosamine. It's just something you have to memorize, just so you know that, whenever they say galactosamine, it's the O-linked one. Okay? For asparagine, since asparagine has nitrogen, like it has a lot of amide nitrogen on its end, it's called N-linked. So the carbohydrate chain attaches to the N, and that's why it's N-linked glycosylation. The substance is called now N-acetyl glucosamine. The only way this can be confusing is because both of them have N, I know, but one of them is galactosamine is for O-linked, and glucosamine is for N-linked. Okay, do we have to know the structure? No, you don't. I'm just making it clear for you guys to understand why is it O-linked here and why is it N-linked here. Okay, but just know that. So asparagine, because it has the nitrogen group, that's why it's N-linked. The rest of the three are O-linked. They're kind of, you can memorize it as like galactosamine, and acetyl galactosamine has a bigger name and it has a lot more amino acids, serine, threonine, hydroxylysine, which are like long names. N-linked is only one example he gave you is asparagine, and it's N-linked glucosamine, which has a shorter name than galactosamine. Other difference in this slide, O-glycosylation happens in Golgi apparatus, which is another organelle, and glycosylation happens in endoplasmic reticulum. This is easy to memorize, kind of, because O, you can memorize it as like Golgi. Golgi has O in it, so that's where O happens, O-linked. Is this clear? Is this slide clear for everyone? Okay, so these changes are, they're much more easier to understand than the whole translation. So this is again phosphorylation. We said glycosylation, 2 types, O and N linked. The last attachment is hydroxylation. This is also easy to understand because hydroxylation is basically you're adding a hydroxyl group or an OH group to the substance. So there are only 2 amino acids that you can add hydroxyl to, which are given here as proline and lysine. I used to memorize it as like they both have L in them, so proline and lysine can have the hydroxyl, which is the L. So proline and lysine are, an example of this is in collagen, alpha chains of collagen. Collagen are things that make up our connective tissues like our skin and so on. And these happen, this hydroxylation of proline and lysine makes them into hydroxyproline and hydroxylysine by these enzymes, again from the name, hydroxylase, vitamin C dependent hydroxylases, they're hydroxylated. So basically this enzyme, hydroxylases, is vitamin C dependent, meaning that this process needs vitamin C like in your diet to happen. And this takes place in the ER as most of the things happen. So you don't need to like really memorize that. Just know that it happens in the endoplasmic reticulum. And the example is collagen in connective tissues such as tendon, cartilage. So if you don't have vitamin C enough in your diet, you end up with a disease called scurvy, which is like weak gums, pale skin, all those, because you're not making enough of collagen, because there's not enough hydroxylation. Is this clear for everyone? Okay, that's good. So we talked about these 3. Last slide of this lecture is trimming. So trimming is from the name, you're cutting a protein, right? An example of that is when a protein is made, but it's inactive. So they are called zymogens. So they're kind of, imagine them that they're like folded proteins that are inactive, but if you cut a part of it, it exposes the needed like amino acid for it to do its function. So once you trim it, once you cut it, it becomes active. So it becomes an active protein. So that's why trimming is an important process too. Yes, active site exposed. One example of that is here on the slide. It's trypsinogen, which is an enzyme in the pancreas. It's not active because if it was always active in our stomach or in the pancreatic juice, it would digest our own cells. But it's only made active when trypsinogen is cut into trypsin. So a part of it is cut off. Yes, when it's part of it is cut off, it becomes trypsin, which is the active form, and it digests the food only, not our cells. So is that clear for everyone? This is like the main thing. Okay. I have a question for you guys if you don't have for me, but that's basically the end of this lecture. This is the first question. There's 3, and like I want you guys to have a break before the one at 7. So let's finish. So a 4-year-old African American girl is brought to her physician by her mother because she has been complaining of episodes of extreme pain, discomfort in her legs and lower back. She appears jaundiced and has a hematocrit of 23% and a hemoglobin level of 7. Her mother reports that she has family members who experience the same symptoms. What is the mutation that causes this presentation? First, what is the presentation? What disease is it? And then what's the mutation? Okay, someone said B, someone said B. Sickle cell anemia, B, sickle cell. Okay. You guys are right. But wait. You guys are right about the sickle cell, so good job. This is the confusion that happened a few slides back, but read B carefully again. Because I would get confused if I saw that. Where did someone Bilal? Yeah. You can see, yeah. There you go. So it's D. I hope this clear, like never makes you forget the difference between glutamine and glutamic acid. Okay. So the answer is, D. I'll explain why. So jaundice is just an example, like it's a presentation that can happen within all these things. But the main symptoms you see here are anemia, which always happens with sickle cell. It's sickle cell anemia, and you have extreme pain and discomfort. So because of all these being stuck in the capillaries, you have a lot of pain. Jaundice just, it can, like, it doesn't have to be always there, but it can be there. Jaundice is when like you have a lot of, okay, wait, it might be too advanced, but you have a lot of bilirubin, like it's not, the liver is not functioning well and so on. Glutamic acid. Okay, so why not B? Because it's glutamic acid or glutamate, as you guys said, not glutamine. This whole sentence is correct. It's replaced by valine at the 6th position. That's fine, but it's not this amino acid. So the answer is D. Yes, glutamine is in Huntington. Good job. So D is none. So D is the correct answer, and I have the answer in all the notes. Discomfort in legs is due to clogging of narrow arteries by sickle-shaped erythrocytes. You're absolutely correct. Do we have to know positions? Yes. That's why I said it's better to know positions because it might be like this. So phenylalanine deletion at 508 position is which one? Which disease? First one is SD. Wait, what do you mean? It's fine, it's fine. You guys know it. Yeah, cystic fibrosis. Yeah, go. So it's not C because it's the wrong position. Okay, second question. Good job on the first one though, because you guys realized what the mistake was. After N-glycosylation, what is the fate of the protein? So ERB is endoplasmic reticulum. It's just, I don't know why there's B in it, but yeah. Okay, someone said A, it stays in the endoplasmic reticulum. So yeah, you guys need to know that N-glycosylation, yeah, it's a modification of the protein, remember? Okay, you guys are saying A. Does anyone have other answers you want to say? Basically, this question is asking, wait, I'll say this. Do you guys have any other answers? C, okay, someone said. Yeah, I know. It's, I think the question was just like that, but it basically means endoplasmic reticulum. RER, rough endoplasmic reticulum. Someone said B, someone said C before. Okay. Okay, one of them is correct, but which one? B. Okay. So the correct answer is B. Yeah. So whoever said B, you're right, and I'll explain why. Because N-glycosylation happens in the rough endoplasmic reticulum, right? But what happens after it? Yeah, after it. So after that modification, where does the protein go? Does it stay in the rough endoplasmic reticulum forever? No, not all of them do. Does it get secreted out of the cell? Yes, all of them get secreted out of the cell because, like I said, all proteins have their own functions. They just go into the rough endoplasmic reticulum to get changed, to get to the final structure, and then they move out. Exocytosis goes to membrane. Yeah, you're right. So C, phosphate group is added. It can be added in the rough endoplasmic reticulum, but not all of them. So it's like not the most correct answer. D, it undergoes O-glycosylation, unlike, because it already went through N-glycosylation, so it's probably something like arginine, which can't go under O. What slide was this on? Wait, so same thing is, yeah, no. Remember what I said? So same, does anyone remember where O-glycosylation happened? Not here. Yeah, Golgi apparatus. So the question would be kind of different. Like it wouldn't, I mean, it would get out of the Golgi, so it's the same answer, yeah. But basically the place is not the same as the end. Someone asked which slide was this on? This slide was here, I think. Yeah, here. But then I meant N-linked happened here in the endoplasmic reticulum. And then what I wanted you guys to know from this is that this is just a place for proteins to be changed. But usually they go out of it. Yeah, but it's not, yeah, it's not written, but from what I said. Here it says, yeah, that's basically it. But you guys get the main idea. Which slide is Okay. Phenylalanine. I have just one more question and we can leave. Okay. Also, which slide is phenylalanine position 6? There isn't any. That's a wrong answer. So the answer for the first one would be this one. If I asked you about cystic fibrosis, someone coming to you with coughing and like clogged airways and so on, that's cystic fibrosis, you would choose A. C is just like a distraction, so like it's not the correct position. So proteins modified in the RER get secreted. Yeah, you're right. So if it's the rough endoplasmic reticulum, it goes out of it to do its own functions. Like it can stay in the cytoplasm, it can move out of the cell, it can go into nucleus, like mitochondria, all the other organelles. So the answer is none of the above. Yes, okay, so I'll send you guys this PowerPoint. All of them have the answers in the notes. This one is none of the above, D. This one is B, because all proteins out of the endoplasmic reticulum after being modified, they go out. They go out of the cell. Um, oh yeah, what I meant by this is, sorry about what I mentioned before, but it gets secreted out of the entire cell because it has its own function. So if it's the nucleus, it goes directly from the ribosome with the nuclear localization signal. You guys remember that? So just know that for this, whatever goes through rough endoplasmic reticulum goes outside of the cell. Okay? Okay. I hope that's clear. So this is the last question. The DNA sequence M shown here is the same is the sense strand from a coding region known to be a mutational hotspot. So there are mutations that can happen in this sequence. It encodes for amino acids 21 to 25. It's not that important. Which of the following sequences is a frameshift mutation that causes termination of the encoded protein? Okay, so which one of these sequences? So this is, by the way, this is here. Wait, I won't say, but basically, what changes can you see? Which option has a stop codon in between? That's what it says. Frameshift. Sorry, frameshift. Okay, someone said A. Deleted G. Okay. Yeah, I know. All of them have the same amount. Okay, which one? I said it by mistake before, but which one of them has the stop codon? Like it gets made into a.
Yeah, see,
Wait, someone said
UAG.
which one?
Does no one get to talk on class? Someone talked before, but usually it's through chat.
Wait,
Yeah, I don't know.
what? Sorry, I can't hear you.
Why did someone say my name?
It's fine. But I heard someone say, yeah, I heard someone say the answer. Okay. Yeah, I know it takes time. It's fine. But basically, you would like. Okay, I'll go over it. Do you guys have just throw answers? It's fine. I'll explain. I'll explain right now. Okay, someone said B. Did I wait for? It's okay, don't worry about it. But I heard someone, like, I heard you realize something, like, from the answer, which is good. Okay, I saw a lot of these. And okay, since I don't want to take a lot of your time, the correct answer is B. Because basically all of these they all have three nucleotides. Like none of it is being like like all of them have a different kind of amino acid, I would say, but none of them causes frame shift, right? But B in general, like B in specific, sorry, has TCA in it, and it's the only stop codon that you see here. So these are DNA sequences. Remember that these are DNA sequences. So when it's done into RNA, this will code for wait. This will code for so wait, let me explain. Just give me 2 minutes, okay? I'll try to finish this up. Yeah, someone said UGA. So here, and if it's RNA, so let's imagine 5 to 3. So RNA would 3 to 5. This would be AGU. So the RNA, the actual sequence, when you flip it to be 3, it's UGA is a stop codon, right? UGA is a stop codon that stops this whole thing. The main thing, again, I wanted you guys to get off of this slide is that since it stops, it kind of changes the whole sequence. Again, like I said, it's a nonsense mutation, right? But it's completely like cuts off the sequence that comes after it. So it can be considered as frame shift. It's unlikely that they'll confuse you with frame shift and so on. Like they might remove this, for example, then it would be frame shift for sure, because now it's a different or for example, like this, so that it's a different kind of sequence. But again, I just wanted to, did you guys understand why it's B? It's the only one with stop codon. How are the options related to the sequence they gave us? Okay, so this is the sequence, right? Now, again, you're not supposed to know what amino acids they each code for, but just know that you can see, just look for a stop codon. That's what I mean, because all of these code for a different amino acid, except the one that's like codes for stop, this one. So, for example, like this is CCC, this is a different amino acid, but it doesn't cause frame shift anyway, you know? I know it's kind of confusing, but do you guys get why it's B? But 3 bases are added. Okay. Yeah, I know. The wording is not that good. But I got this from one of the question banks, I remember. 3 bases are added. Not really, they're just changed. All of them are changed. Like none of them are exactly the same as this sequence. Okay? So, okay, I don't want you to get confused, so you can ignore this slide if you want to. Just if I say which one has a nonsense mutation, you guys will all know which one it is, right? Because so you just have to flip into mRNA. What does that mean? The question before I don't know what that means. If you can translate that for me. Okay. Okay, yeah, sure, I don't mind. This one, you mean? Okay, again, okay. So I hope it's clear for everyone. I know the wording is not that good, but I copied it off from a resource bank. So that's just the explanation for it. So imagine the question, well, they won't ask you this for sure, I think. So just know that the stop, like if I say what's the stop codon, which of them has nonsense mutation, it's this one. Okay, just know how to convert it. For this question, after N-glycosylation that happens in the endoplasmic reticulum, what happens to the protein? It goes out of the cell. all of it goes out of the cell. It doesn't stay forever because it has to do its function, right? A phosphate group is added, it still means that like this still happens in the plasmic reticulum. So not all of them will have phosphate group added. It's only serine, threonine, tyrosine. It undergoes O-glycosylation, unlikely because it's N. So the only correct answer here, you can see that it's B. Like you can eliminate the rest. Is that clear for everyone? I'm sorry if the last question confused you, but okay, no problem. But I just know how to convert it. That's the main idea, okay? Inshallah. Okay. If you guys have any more questions, feel free to like contact me. I don't mind answering anything. And yeah, you guys, I know you have a lecture at 7, so it's okay if you want to leave. I thought, wait, there was one question. I thought the mutations just happened to the mRNA. Oh no, the nonsense, missense, and the silent happens to the DNA. So the mistake in the DNA is like translated into the mRNA, right? So the mRNA reads like the whatever mistake is there in DNA. What do we have at 7? There's one by Aris, if I'm not wrong. Did they change it? They have one. He has one today about epigen, like what was it? Gene regulation. Oh, that's tomorrow. Oh, okay. I thought I was rushing for that. Okay, good. That's good. Transcription 1 and 2 at 7. Okay. Yeah. No, I don't. I can check. I can check the schedule for you if you want. But no problem. Yeah, it is tomorrow. It is tomorrow. I thought it was just today. That's why. No problem. I'll stay for 1 minute if you guys have any questions. But other than that, yeah, I'm done. No problem. You guys are so sweet. Thank you. And I'm really proud of everyone because for attending or